The fingerprints left at the scene of crime are latent prints. Latent means hidden, which also means that we are not in a position to see them. When a finger touches a surface, it leaves behind a deposition of sweat exactly in the same pattern as is there on our fingertips. But we can't see it because sweat is colorless. That's why it's called a latent fingerprint or a hidden fingerprint. In order to detect it, we have to develop it. There are many chemical reagents which develop the latent fingerprints. The fingerprint residue contains a host of chemicals. Amongst these, amino acids are most important. If any reagent reacts with amino acid content of the sweat, it makes them colored. We say that the fingerprint has been detected or the fingerprint has been developed. The most important chemical which reacts with the amino acid content of sweat is Ninhydrin. It gives a purple colored complex called Ruhmann's purple. Ruhmann was the scientist who first carried out this reaction, and purple, of course, is the color. Now you can imagine the amino acids in our sweat were colorless, and by a simple chemical reaction, we have converted it to purple colored compound. So whatever was not visible earlier, it becomes visualized, and it gives very sharp prints. Amino acids actually do not migrate within the capillaries of the documents and surfaces. So very old prints can also be developed with the aid of Ninhydrin. Of course, some parameters have to be adjusted, like temperature, humidity. They should be optimum before development starts. One drawback of this method is that the complex which is formed, which we call Ruhmann's purple, is not fluorescent in nature. So that sometimes weak and faint fingerprints cannot be detected appropriately. However, Ninhydrin is not the only method of detection. There are several reagents which give fluorescent compounds with amino acids. The reaction is very simple, and after the reaction has been carried out, the developed print is impinged with ultraviolet light or with laser light, and those weak and fragmented fingerprints, they become enhanced, and a good photograph can be taken. Apart from Ninhydrin, now we have several analogues of Ninhydrin which can be used as and when required. The analogues can be easily prepared, and some of them are now commercially available in the market as well. The method is cost effective, and very old fingerprints, sometimes 5 to 10 years old also, can be developed by this method.
After studying this module, you shall be able to know that amino acids are an important and significant constituent of set residue. There are a number of reagents that fix amino acid constituents of sweat to develop latent fingerprints. Some of the products formed by the interaction of developing reagents with amino acids are fluorescent in nature. In some cases, fluorescent products may be produced along the ridges after appropriate post treatment, and you'll know that a number of amino acid specific reagents are used for detecting latent fingerprints in actual casework investigation. Let us start with the introduction. Amino acids constitute an important constituent of fingertip sweat. When a finger touches the surface, these analogues are deposited thereon as sweat residue. This deposition, called latent fingerprint, contains several other chemicals such as chloride, phosphate, sodium, potassium, and ions, as well as fatty acids, carbohydrates, and vitamins. However, unlike other sweat constituents, amino acids neither interact with the cellulose component of paper, cardboard, wood, and other porous surfaces, nor do they migrate within the substrate. As a result, the reagents that give color derivatives on interaction with amino acids render visibility to both fresh and old fingerprints on a wide array of surfaces. It is therefore not surprising that amino acid specific reagents have been the state of art strategy for paper exhibits for the past many decades. Let me tell you something about amino acid reagents. The amino acid components of sweat may be tagged with a number of reagents. The first, and for many years the only amino acid based reagent that was standardized, was Ninhydrin. For this reason, it has been the most popular one. Over the years, many more amino acid specific reagents have been investigated for detecting latent fingerprints. We review their significance in this module. Let us start with Ninhydrin. Ninhydrin treatment is one of the premier techniques for development of the latent prints on porous surfaces such as paper. Ninhydrin reacts with amino acid content of sweat, forming a purple complex called Reuman's purple. Broadly, the procedure for developing latent finger marks involves spraying the ninhydrin solution on the surface containing the impression from a distance of about 6 inches. After the solvent evaporates, the solution is sprayed again. The surface is then heated for a short time without allowing it to come in contact with the heat source. If quick development is not required, the item may be allowed to dry at room temperature. Better results are obtained by natural drying. A sample fingerprint developed by Ninhydrin method is shown in figure. However, the concentration of amino acids in sweat is quite low, as a result of which the developed prints do not show sharp contrast. To improve their clarity, prints are post-treated with salts of group 12 elements of the periodic table. This results in conversion of non-fluorescent Reuman's purple into a photoluminescent complexes. The color of ninhydrin developed fingerprints changes to red or orange on post-treatment with zinc chloride or cadmium nitrate. Subsequent observation under an argon ion laser produces appreciable luminescence. The intensity of photoluminescence increases at low temperatures. Now, let us study about ninhydrin analogues. Till the early 1980s, it was endeavored to improve the ninhydrin method by altering the solvents, the composition, or by pre-treatment or post-treatment of developed prints. Later, a different approach was adopted. Several derivatives of ninhydrin were synthesized and tried out for developing latent prints. It was anticipated that the derivatives of ninhydrin possessing the diketone function would react with amino acid content of sweat in the same way as the ninhydrin itself. With several ninhydrin analogues, this expectation turned out to be true. The most promising ninhydrin analog is benzoninhydrin. This reagent develops dark green colored fingerprints with excellent resolution. Improved sharpness was observed on treating benzoninhydrin developed prints with zinc chloride, following by examination under an argon laser. However, still better results were obtained with the frequency doubled, neodymium is to yttrium aluminum garnet laser. The emission wavelength of the later device closely matched with the absorption band of the benzoninhydrin amino acid zinc chloride complex. Another ninhydrin derivative which develops latent fingerprints with appreciable degree of sensitivity is 5-methoxyninhydrin. On paper, this reagent produces fingerprints as efficiently as ninhydrin, yet fluorescence after zinc chloride treatment is considerably strong than with ninhydrin, particularly when examination is carried out by a copper vapor laser. 5-methoxyninhydrin has also proved useful for developing fingerprints on counterfeit currencies. Another analog is 1,8-diazafluorine 1,8-diazafluorine is also known as DFO. It is an amino acid sensitive reagent which develops latent fingerprints on porous surfaces like paper and wood. On reacting with the fingerprint residue, it gives a pale pinkish purple luminescent product. The test solution is prepared by dissolving 0.5 gram DFO in 1 liter of methanol or petroleum ether. The surface bearing the latent fingerprint impression is immersed in the reagent for 5 seconds, dried, re-immersed for 5 seconds and heated at 110 degrees Celsius for 10 minutes. Under white light, pink-purple ridges are rendered visible. The surface is illuminated with radiation of 470 nanometer wavelength and fluorescence is observed through a 570 to 600 nanometer filter. The luminescence is at its maximum immediately after the heat treatment and then decreases slightly with time due to absorption of ambient humidity. The luminescence may be restored to its original intensity by reheating the prints. Fourth analog is fluoroscamine. It is a spiro compound that is not fluorescent itself but reacts with amino acids to form highly fluorescent products. Hence, it is being used as a reagent for the detection of amino acids and peptides. It is also a fingerprint development reagent. A solution of 20 mg fluoroscamine and 0.5 ml triethylamine in 100 ml acetone is sprayed on the surfaces containing the finger mark. The surface is dried and observed under an argon laser. Sharp and clear fingerprints can be rendered visible. Fifth is teufelaldehyde. Teufelaldehyde reagent, the structure of which is set out in figure which is given, is used as a very sensitive fluorescent reagent for assaying amines or sulfur hydride in solution, notably contained in proteins, peptides, and amino acids. As a result, it also reacts with amino acid and protein components of sweat residue to reveal latent fingerprints. On examination under an ultraviolet lamp or an argon laser, the developed prints are enhanced due to fluorescence. Two solutions are required when 2-phenaldehyde is to be used as a fingerprints reagent. Solution A is prepared by dissolving 2.5 gram boric acid in 95 ml distilled water. The pH of the solution is adjusted at 10.6 with 6 molar potassium hydroxide. To this solution, 0.3 ml of 30% detergent and 0.2 ml 2-mercaptoethanol is added. Solution B is prepared by dissolving 200 mg Teufelaldehyde in 2 ml methanol. Solution B is mixed with solution A. The mixture is then sprayed on the exhibit. On drying, the surface is examined either under an ultraviolet lamp or under an argon laser, whereupon fingerprints are rendered visible. Sixth is Dansyl chloride. Dansyl chloride, the structure of which is depicted in the figure, reacts with primary or secondary amines, amino acids, and proteins to produce stable blue or blue-green fluorescent sulfonamide adducts. It is widely used to determine protein sequencing and for amino acid analysis. It may also be used to label hydroxyl and carboxylic acid functional groups. In forensic applications, it is a fingerprint reagent for detecting latent fingerprints. The composition is prepared by dissolving 10 mg dansyl chloride in 20 ml acetone. The solution is diluted by adding 80 ml freon. The test solution is sprayed onto the surface and allowed to dry. Clear fingerprints can be observed upon ultraviolet illumination. 4-dimethyl aminocinnamaldehyde. The structure of 4-dimethyl cinnamaldehyde is shown in the given figure. The test solution is prepared by dissolving a mixture of 4-dimethyl aminocinnamaldehyde and sulfuric acid in ethanol. The solution is further diluted by adding ether. The mixture is sprayed on the article and on drying the fingerprints may be observed under ultraviolet or laser light. The background coloration, if any, may be reduced by keeping the developed prints in the dark for a few days. The reagent is, however, not good at developing old prints. 4-dimethyl aminocinnamaldehyde may also be used as a fumigant for visualizing fingerprints on a wide range of surfaces. Another kind of amino acid reagent is 4-chloro-7-nitrobenzo 2-oxa-1,3-diazole. The 4-chloro-7-nitrobenzo 2-oxa-1,3-diazole is used for fluorogenic detection of latent fingerprints on multicolored paper. About 0.1 gram 4-chloro-7-nitrobenzo 2-oxa-1,3-diazole is dissolved in a mixture of 10 ml acetone and 90 ml fluoresol. Colored paper is dipped in the solution for a few seconds, air dried, and heated at approximately 80 degrees Celsius for 2 minutes. The developed prints are examined with blue light and observed through a 580 nanometer orange filter in a dark room. Another reagent is 8-aniline naphthalene 1 sulfonate. It has proved to be a valuable and sensitive reagent for detection of fingerprints on greasy and porous surfaces. The developed fingerprints, when impinged with long wavelength ultraviolet light, show bright yellow fluorescence against dark background. Rapid development, no heat requirement, and simplicity are some of the other advantages of this technique. About 1 gram of 8-aniline naphthalene 1 sulfonate is dissolved in 5 ml methanol and the resulting solution is mixed with 200 ml freon. After the two layers separate out, molecular sieves are added to remove methanol as it causes running of ink on scripted documents. After spraying the test solution on sample fingerprints, the surface is allowed to dry out for about 1 minute. It is then examined under long wavelength ultraviolet light, whereupon fingerprints are rendered visible. Next is 7-nitro 4-benzofuranosyl ether. 7-nitro 4-benzofuranosyl ethers are fluorogenic reagents that can develop latent fingerprints on paper with a high degree of sensitivity. These may be used both in solution form as well as in vapor phase. Although the development is faster when a solution of the reagent is used, in vapor phase, detection there is a smaller background fluorescence and discoloration. The most important compound of this classification, which is mostly used for latent fingerprint detection, is 4-methoxy 7-nitrobenzofurane. Its structure is shown in the figure. The spray solution is prepared by dissolving 100 mg of 4-methoxy 7-nitrobenzofurane in 1 ml acetonitrile and fluoroceol diluted in 100 ml with samples bearing latent fingerprints are dipped into the test solution for 15 seconds and then air dried. These are then heated in an oven to 110 to 120 degrees Celsius for 10 to 15 minutes. Clear and sharp fingerprints are developed. For vapor phase application, about 20 mg crystals of the compound are placed in a glass beaker and fingerprint samples are suspended about 15 cm from the base of the container. The beaker is covered with a thin aluminum foil and heated in an oven to 60 to 90 degrees Celsius for a period of 1 to 7.5 hours, whereupon the fingerprints are visualized. 11th reagent is 1,2-indanidone. The reagent 1,2-indanidone develops excellent quality fingerprints on porous items by reacting with amino acid content of sweat residue. It is specifically useful for detecting prints on bank check and thermal paper. Its structure is depicted in the given figure. The test solution of the reagent is prepared by dissolving 2 gram 1,2-indandione in 70 ml ethyl acetate. The contents are diluted to 1 liter by adding 930 ml petroleum ether. The solution is sprayed over the article bearing latent fingerprints and then heated in an oven for 20 minutes at 100 degrees Celsius. The fingerprints so developed are illuminated with light of 515 nanometer wavelength and observed through orange goggles. Finally, these are photographed. Now, let us summarize this chapter. So, in this chapter, we have learned that latent fingerprints may be visualized by a lesion of amino acid specific reagents over the years. Ninhydrin has been branded as the most efficient amino acids tagging compound as far as detection of latent fingerprint is concerned. However, the reaction product of ninhydrin and amino acids is non-fluorescent and only after post-treatment yields luminescent product. The other amino acid specific reagents are advantageous in that these products produce fluorescent derivatives on reacting with amino acid content of sweat residue in one step. Two of these reagents, 1,2-indiandione and 1,8-diazofluorine 9,1, have been validated for detecting latent fingerprints on porous surfaces. The detection methodologies have been standardized and these procedures are being used in casework investigations.